Hi, today I would like to show you how to use Claude Code with genomic intelligence models to solve biomedical tasks and develop new drugs. Recently, a company called Genomic announced almost $100 million rise for a new type of genetic drugs that can cure the cancer cells. This drug is based on gene therapy, and the gene, in this case, is a toxic gene that can kill the cell. It is used to kill cancer cells, but what is important that the gene is controlled by a specific DNA sequence called promoter. It makes the gene active only in cancer cells, but not in normal cells. That's very important because the drug targets glioblastoma cells located in brain. So if the gene will be damaging healthy neurons, the therapy will cause too much collateral damage and can be used. It's tricky to make the sequence that will activate gene only in cancer cells, and it took the company a couple of years to develop this DNA switch. It was lots of candidate probing in laboratory, and right now I'm going to show you how we can speed up this cycle significantly using the Claude Code with genomic intelligence models. So we prompt Claude Code with a specific text describing what we would like to achieve. So we say we want a regulatory sequence with high predicted expression in glioblastoma stem-like cells and low predicted expression or activity in normal brain. We explain how to use Genomic Intelligence API, and we explain biological details about where the drug should be active and where it should not damage cells. So let's start the code now. It will read the API and available models. So once it has read the documents, it will start from downloading data and drafting a pipeline that is able to design the sequence that can control toxic gene in silico. Now we have the workflow ready, and we are running 100 cycles of DNA sequence optimization, which would take months or even years in a lab, and which would take just a couple of minutes using the models embedded in Claude Code. So after 60 cycles of optimization, we have already increased the selectivity 2 times, almost 3 times actually, and we have now target expression much higher than off-target expression. With these candidates, we can already go to the laboratory and test them, and we can continue these cycles of optimization more and more to get more promising candidates for drugs. Okay, so the results are ready now, and in just a couple of minutes, we have a new candidate promoters, which was designed by starting with some plausible candidate from human genome, and then with 100 cycles of optimization that will took months or years in a laboratory, we were able to increase the selectivity of this DNA sequence a couple of times, which means that we now have a very good candidate for laboratory validation. And instead of running cycles of this validation, we can go with the right candidate directly into the wet lab and then use the right sequence as a drug against the glioblastoma. So that's how the genomic intelligence models that can predict the DNA grammar together with a Claude Code that can organize the workflows and use these models in real biological settings together speed up the development of new drugs from years into minutes.